All right, let's dive right into this explainer. Today, we're unpacking what is absolutely the most devastating biological heist in human history. We're talking about an organism that literally reshaped the geopolitical map, completely took down empires, and profoundly altered the course of human history. But, we aren't just going to look at the history books here. We're actually going under the microscope to see exactly how this pathogen pulls off its deadly infiltration.
Just imagine for a second if the bubonic plague hit the US today. Based on the historical mortality rates from the mid-14th century, a single microscopic organism would wipe out somewhere between 110 and 150 million people in just a couple of years. I mean, that's nearly half the population just gone. The sheer scale of that kind of destruction is honestly almost impossible to wrap your head around, which is exactly why understanding this organism is so absolutely crucial.
Part 1. A history of devastation. So before we zoom into the actual microscopic warfare, we really have to grasp the massive geopolitical destruction this single bacterium has caused. You see, the plague didn't just strike once and disappear. No, it came in relentless, civilization-altering waves. First in 541 AD, completely laying waste to the last resurgence of Roman power under Emperor Justinian, killing up to 10,000 people a day in Constantinople. Then we have the infamous 1347 Black Death, which killed so many people that historians actually argue it forced the transition from the Middle Ages into the Renaissance and the modern era. And finally, the third pandemic, exploding out of Hong Kong in the late 19th century and claiming over 10 million lives in India alone. But you might be wondering, how did that infamous 1347 pandemic actually reach Europe? Well, it didn't just appear out of thin air. It was arguably a terrifying early act of biological warfare. Get this, during the 1345 Mongol siege of the city of Kaffa, the Mongol army itself became infected, and as they were dying, they reportedly used catapults to hurl their rotting, plague-infected corpses right over the city walls. Yeah, literal biological weapons. The terrorized Genoan sailors who were defending the city eventually fled by ship back to Europe, completely unwittingly carrying the horrifying disease right into the ports of Italy. Part 2. Unmasking the microscopic killer. It wasn't until the third pandemic that scientists Alexander Yersin and Kitasato Shivasaburo finally unmasked this killer, a bacterium eventually named Yersinia pestis. Now, Yersinia pestis is a textbook example of zoonosis. Around 10,000 years ago, it evolved from a related gastrointestinal animal bacterium, but it perfectly adapted its genetic code to jump from wild rodents and fleas directly into us, into humans. Seriously, it is a dark evolutionary masterpiece of cross-species transmission. Which brings us right to the core mystery of our explainer today. To truly understand a death tolls in the hundreds of millions, we have to ask, how on earth does this tiny pathogen pull off the ultimate cellular heist? I mean, how does it enter our bodies and completely dismantle our incredibly sophisticated innate immune system? Let's take a look. Part 3. The flea's starvation strategy. So, zooming right into the gut of a rat flea, where this invasion actually begins, the bacteria literally weaponize the flea's hunger. It's crazy. Inside the insect, Yersinia pestis produces a biofilm that completely blocks the flea's digestive tract. The flea actually feels like it's starving to death, which forces it into a total feeding frenzy. It violently feeds on a mammal, like a human, but because its gut is blocked, it can't swallow. Instead, it regurgitates the sucked blood, along with a massive payload of those blocking bacteria, directly into the skin of its victim. The heist has officially begun. Part 4. Hijacking the immune system. Okay, so we've breached the skin, and now the pathogen is met by the body's first responders. And this is exactly where the sheer, evil genius of Yersinia pestis really shines. See, when it faces a neutrophil, it's a death trap. The neutrophil just gobbles the bacteria right up and digests them, no problem. But when it faces a macrophage, things change completely. Macrophages are supposed to engulf and destroy invaders in a digestive vacuole called a lysosome, but Yersinia pestis brilliantly flips the script. Using special outer proteins, it stops the lysosome from merging with it. So instead of being digested, the bacteria use the macrophage as a safe, cozy little incubator to replicate. It's literally like a burglar stealing a police cruiser to ride safely straight into the police station, which, in the human body, is our lymph nodes. And while they're chilling inside that hijacked macrophage, the bacteria don their disguises. They produce a pilus called the pH6 antigen that binds to our body's own LDL, what we normally call bad cholesterol, so our immune system just sees a normal cholesterol carrier floating by. Nothing to see here. They also wrap themselves in an F1 antigen capsule to resist being eaten by other immune cells. And most incredibly, they alter their own cellular structure. Normally, bacteria have an outer layer called LPS with 6 acetyl groups, which basically triggers super loud alarms from our toll-like receptor 4. But Yersinia? It changes its LPS to have only 4 acetyl groups, effectively silencing the alarm and slipping right past our security sensors.
Part 5. Disarming the body's defenses.
So once our hijacked macrophage safely delivers these cloaked bacteria to the lymph nodes, the real heavy sabotage begins. Yersinia pestis deploys a terrifying weapon, the YOPs, or Yersinia outer proteins. Just think of these as microscopic needle-like syringes. The bacteria attach to our immune cells and literally inject specialized biochemical weapons directly into the host cells. Their goal? To paralyze the guards and cut the communication lines. I mean, this is a highly coordinated heist. First up, YOP-M destroys our natural killer cells, drastically reducing our ability to even fight back. Then, YOP-P and YOP-J take away the chemical hooks inside our blood vessels. Why? So emergency neutrophil backup forces just slide on by. They can't even enter the infected area. And if that wasn't enough, the LEL-CRV protein completely mutes toll-like receptor 2. That means the crucial inflammatory cytokines, which are essentially the body's emergency sirens, are never turned on. It is a total 100% communications blackout.
And for the grand finale, as a final sabotage step, Yersinia pestis uses something called the ALE protein. You see, our blood has this complex, cascade-like backup defense called the complement system, which usually ends in a potent microbe-killing force called C3 convertase. Well, the ALE protein just steps right in and deactivates the C4 component, breaking the entire chain. Boom. The body's ultimate backup weapon is effectively dismantled before it can even fire a single shot. Part 6. Symptoms and modern threats. All right, zooming back out from the microscopic level, we start to see the horrific physical consequences of all this unchecked replication. Because by the time the immune system finally realizes what's happening, it's way too late. The bacteria have multiplied into the millions. That massive delayed immune response triggers a devastating cytokine storm. Your lymph nodes become packed with dead bacteria and immune cells, swelling to the size of actual eggs. These are the infamous buboes, incredibly painful and prominent in areas like the groin or armpit. And as that cytokine storm rages on, blood clots form in the vessels, completely cutting off oxygen and leading to blackened, necrotic tissue on the fingers and toes. It's a horrifying symptom, and honestly, it's likely what gave the Black Death its name. If left unchecked, the disease progresses terrifyingly fast. It moves from that localized swelling in the bubonic phase to septicemia, which is where the bacteria pour directly into your bloodstream. And from the blood, it can easily reach the lungs, becoming pneumonic plague. Now, at this point, it is incredibly dangerous. The victim can simply cough the bacteria into the air, infecting others around them without a flea ever being involved. And untreated, pneumonic plague usually results in death in just a day or two. Now, you might be sitting there thinking, well, okay, but we have antibiotics now, so this is all just ancient history, right? Not quite. Yersinia pestis belongs to a bacterial family that is absolutely notorious for developing antibiotic resistance. In fact, in 1995 down in Madagascar, doctors isolated a strain from a 16-year-old boy that was resistant to 6 major antibiotics. 6. Because of its extreme deadliness and its ability to rapidly acquire resistance genes, it remains a highly potent bioterrorism threat even today. So, knowing how brilliantly this microscopic mastermind dismantles our defenses, cloaking itself, hijacking our cells, and systematically cutting our immune communication lines, we really have to ask ourselves a very unsettling question. With its ancient blueprint for cellular hijacking still perfectly intact and its proven ability to mutate, is our modern medicine truly ready for its next heist? Something to think about. Thanks so much for joining me on this explainer, and I'll catch you next time.